So after transcription, you have an mRNA molecule that contains the introns, which are your non-coding regions. Well, that is if it's in a eukaryote. So that means that these non-coding regions have to be removed, and this is through something called splicing. As well as this, some things are done to protect the mRNA molecule before it can be translated into a protein. So these things to protect the ends are called capping and polyadenylation. So your capping is when you have a methyl group added to the 5' end, and then your polyadenylation is when at the 3' end you've got an addition of around 150 to 350 bases. These bases are your adenine bases. This stops the mRNA being degraded by exonucleases, and it also promotes the ribosome binding to it, and it regulates nuclear export of it to the cytoplasm for translation. Another type of post-transcriptional modification is alternative splicing. This is when it can skip exons or actually splice out exons to make a variety of different combinations of them. This means that you get a wider variety of proteins, and it's what makes your eukaryotes so complex.